Cool. So as you saw from my speaker notes, I am Damon. I'm an engineer at Benchling. And so today, life scientists are tackling some of the world's hardest problems. From anything cancer, ALS, these horrible diseases that have a huge impact on everyone, but they're using tools that tend more towards getting in their way than actually enabling them. Benchling's mission is to build world-class tools for collaboration and scientific research and can kind of streamline and automate these workflows. So today, I'm really excited to talk to you about the data warehouse that we've built. So to give you a little perspective, I'll talk first about what Benchling offers as a platform. Then I'll go into our vision for what the data warehouse means. Next, what characteristics we needed as we were designing the system and how we built that. After that, some challenges. And finally, I'll wrap up with some closing thoughts. So Benchling has already made some really great strides in improving research. Imagine a scientist. She's performing some kind of experiment on a bacterial strain. She'll probably try a bunch of different variations and then see how each of those perform on some kind of assay. She wants to know across many experiments in many different test tubes which one of these strains performs best. Unfortunately, this question is actually pretty difficult to answer. The steps that she followed in her experiment either live on paper or in some kind of Word document. The results of her experiment live across multiple spreadsheets. Information about the test tubes that were in those experiments are in different spreadsheets. And information about those strains are in entirely different spreadsheets. So it takes her 6 hours to answer a 30-minute question. So what Benchling offers is a lab notebook, DNA design tools, and a biological repository. What this allows the scientist to do is easily reference information about her experiments, the test tubes she's using in them, and the strains in those test tubes as she's performing her work. But until recently, there's kind of been something missing here. Our lab notebook allows you to reference data that you've captured in a single experiment, but there's no way to compare across experiments. So scientists have to copy data back out of Benchling into spreadsheets, compile these master spreadsheets, and then run their analyses. And then we end up with the same data headache that we set out to prevent. So Benchling has all of this data, but we're not presenting it to scientists in the way that they need. And this is where the data warehouse comes in. By streaming all of Benchling's data to a fully managed database, we're giving them full access to everything we know about their research. The database has, for example, a table for their sequences, a table for their test tubes, a table for all of their quantitative results. And then they can use their favorite analysis tools to connect to this and then run the analyses that they care about. Moreover, our large-scale customers are actually able to leverage this in a really cool way. They can connect their instruments to automatically send data to Benchling, which in turn syncs it to the warehouse. And then from there, they can run custom analyses, data processing pipelines, and even full-blown web applications on top of organization-wide data. So how do we build it? Traditional data warehouses operate on a pretty slow time frame. Typically, they'll sync once a day or maybe once every 6 hours at best. But that's not okay for us. We want scientists to be able to put data into their notebook and then immediately analyze that and compare it to data across the entire organization. We also need to be able to handle large workloads without collapsing. And then failures at any point in the system should never cause an update on Benchling to get lost. So it'll be useful here to consider a concrete example. Benchling allows defining custom data types, and this has some pretty big implications. It means that whether your vertical is manufacturing antibodies or optimizing yeast, you can customize Benchling to suit your needs. In this example, the customers defined a promoter and a protein that they care about on top of Benchling's base sequence. This is represented in Benchling as two separate model types. Firstly, a DNA sequence that has bases, and then secondly, a custom field key-value pair that links to the sequence that it lives on. So let's use that example to see how data syncs from Benchling to the warehouse. We've built a system that monitors changes in our main Postgres database, transforms these changes, and then syncs those to the warehouse. So first, the user makes an update. We automatically record those changes as these events you see in the top right. We recognize which models need to be updated in the warehouse, map that back to what data we think should live in the warehouse, and then finally sync that. So let's start with Postgres triggers. A Postgres trigger is a way for you to tell the database that you care about operations on a table. So here, we've defined a custom function that records the table name, the ID, and the operation into our own event log. So if a user updates a field on their DNA sequence, then our trigger automatically records this update. For each data type that we sync, we've defined a set of queries that, given some events, returns a set of records that need to get synced. So here, we run this query to say, what sequences should I update, given that this custom field has changed? Now, we know that a DNA sequence has been changed. What do we do next? We use what we call mappers to map the DNA sequence into a list of key value pairs that we want the database to show. So this is represented as a Python dictionary, and what's nice about this is we can actually dynamically generate what we want to show up. So here, we don't actually know ahead of time what the custom fields are, but thanks to our mappers, we can just say, this custom field has this name, and it'll get translated into a dictionary, which our system automatically resyncs to the warehouse. Something to note here is that we're not using the DNA sequence at the time of the event, but actually at the time we're processing it. And this is a couple important things that I'll get back to in a second. So now we've got some Python dictionaries, and we want to sync these to the warehouse. We can run a single SQL update command that syncs the warehouse with the application data, just like you're seeing here. Furthermore, based on the table layout that we're noticing from this, we can automatically run live migrations on the warehouse tables. So as the user is updating their custom types to match their research needs, we're automatically syncing the warehouse. So far, we support Postgres and Redshift, but it's actually not that far of a leap to extend to pretty much any kind of data store. So let's revisit those design goals. Usually it's pretty quick. In the simplest case, for a single event, we run query, and we see what records need to be updated. Then we run another query and say, here's what the warehouse looks like. It's also scalable. So we only need to add more workers to process events on both sides. We also get a lot of benefits from the item potency that I just mentioned. If we have a bunch of events on the application, or if we have a bunch of things that need to be resynced, we can condense these all into one update, because we just need to update it once and it'll reflect the current state. And finally, it's failure tolerant. If one of our tasks fail, we can just process it again. If things go super wrong, we just queue up the entire table and it'll resync. And I want to highlight some of the challenges that we addressed along the way. So what happens if a custom field is deleted? By the time we're processing this event, we actually have no idea what DNA sequence it belonged to, because by the time we process it, the data's gone. Our solution is to add another event type here that gets inserted for everything the custom field points to. At the trigger level, we look at all the foreign keys on the record and then add a child delete record to anything that it points to. From there, we can say, this sequence's child got deleted, we should just resync it. Now, because our customers border on clinical studies, it's actually legally required for only subsets of users to see specific data. To do this, we've made use of Postgres' row-level security features. So we sync permission metadata that we can join on so that for any row in the warehouse, we can perform a query to pre-filter what the user should see. This permission metadata maps directly back to our application so that administrators only need to manage one set of ACLs. Row-level security gets us about 90% of the way there, and then there are a couple more sandboxing techniques like removing the ability to create functions and things like that that basically make us have a really secure warehouse. Now, as I mentioned, our system supports an event log as a stream, and that's really, really nice for a lot of things. But one of the places that kind of lets us down a little bit is when we're processing a large transaction in our application database, that will actually get processed in chunks to the warehouse. So the warehouse at any given time might actually be reflecting partial updates. We've got plans to fix this later down the line, basically by recording a transaction ID, and then we can batch all of those records to be synced at once. So, last couple of thoughts. What's exciting is that this is really only the beginning. We've started by syncing all of Benchling's data, and this really does enable a lot of really cool analyses like I've talked about. But it's also not a big leap to say that we could just ingest data from all the on-premises systems, all the instruments, anything that a scientist wants to use. We're also looking into larger scales of data. Our streaming approach works really well for incremental updates, but we don't want a massive gigabyte of scientific data to block the entire stream. At the end of the day, we're a tooling company. We're letting scientists do their job better by putting their data back in their hands. The data warehouse enables scientists to easily perform cross-experiment analyses that have historically required a lot of tedious labor. Far more importantly, though, our warehouse exposes the Benchling backend to large organizations that can do some really, really exciting things with this. Large pharmaceutical and biotech companies have tons of instruments that spit out data, or data processing pipelines that skyrocket in power with an organization-wide centralized data store. And lastly, we're hiring. So if you want to work on some really cool problems in a space that really needs technology's help, I'd love to hear from you. Thank you.
Thanks. So we're going to do questions again, but I'm just going to pick and choose. And could you please repeat the question after someone asks something? Yeah. So are there any questions for Daniel?
The last one. So I've seen there are a lot of similarities in this to other kind of data streaming problems that I've seen. I'd be interested to know kind of what some of the specific, and you highlight a few of them, specific needs for scientific use case are, and also kind of what some of the assumptions you had about the system design going in that turned out to be wrong, that you ended up adjusting to show us the good system that you came up with in the end.
Sure. So the question was two parts. The first one, the most recent part, was some assumptions we had that turned out to be wrong. And the second part is how this compares to other systems. Is that correct?
Yeah, I guess they're kind of, maybe in some sense related, like what's the specific need of the scientific.
Okay, so how does science play a role in this?
Yeah.
So how this compares to, so one of the big things actually that we got wrong early on is we started out with the pretty classic denormalized approach. So a lot of traditional warehouses will have like a fact table and a dimension table, and it's just kind of like spread out everywhere. We thought this was nice, but actually what our users want is a more standard application database. And so this leads to something more like a sequence table and a user table, as opposed to kind of inlining the user email everywhere. So that was one of the big things. Sorry, the other question was how this compares. Why don't I follow up offline with you after that? It's just a very deep topic.
Any other questions?
So I'm working in physics. I'm sort of wondering how these same tools could be applied to something like chemistry or physics, or like what other areas of science have you thought about, and like why is biology the natural starting point?
Yeah, I can't answer why biology is a natural starting point here, but I can tell you that there's a lot of stuff still in biology, and we're really interested in like just kind of owning this one single space.
Yeah.
There are some pretty easy comparisons, like having a physics notebook where you track all your data, and that gets synced to a place where you can analyze all of your data.
Yeah.
But I think how this data relates to like metadata or biological things with custom fields that you want to track, as opposed to a physics problem where maybe we actually should be giving you more kind of powerful like chemistry tools or like analyses, like harder computations that we don't support.
Okay. In the back, there was a question?
Yeah. So I guess you mentioned that you have an RDS and Postgres like tables, right, that you're storing this data in. Like how? So you also offer an API, right, that they're reading all of this data from. Like, is there any kind of like I guess for this particular use case where it is with biologists using like the system, how have you kind of structured that with them reading out the data?
Sorry, can you
How's
repeat?
the API structured, and like how is that particularly structured for like the scientists that are then building interfaces on top of the data that you're storing?
So actually we do expose these databases like directly to our scientists, so they can just connect through a SQL shell and run SQL queries. From there, I think it's kind of up to, like, a lot of these large organizations actually have their own IT staff, and so they are having to build these giant applications. And we're just taking a large chunk of their work out by saying, like, don't worry about storing this data, don't worry about like, managing your own databases or anything like that. We've got that. You focus on your core research and building the science part that you know how to do best.
We have time for one more. Oh, wow, one person shot their hand up, and then we're gonna do these two. Here's
Okay. When you talk about storing data, are you storing it on your cloud services, or are you storing it on the on-premise? And if it's on cloud, do they have any like data ownership issues, like in having someone else hold the data?
Yeah, it's a great question. So the question was, We do store it in the cloud. So the question is basically, do these large organizations have issues with us taking their data? And that actually has been a kind of fighting battle that we've been doing since the company was founded. In general, we have been kind of winning this. They are getting more and more comfortable with letting their data live in the cloud. But in some cases, there are kind of exceptions where even these like super large organizations actually want to own their entire infrastructure set. And so we actually have to figure out how to like build our application on top of their machines.
So how much of your data is like only solely user generated versus like, let's say like publicly available like genomic data sets? Are you guys integrating that, or can I say, oh, I have this data set that I sequence myself? Can I compare that, bring that in from like with a publicly available library?
Yeah, that's a great question. Basically, what does Benchling offer in the way of publicly available data sets? So Benchling as an application does allow you to import genomes, and you can use those genomes and the annotations that come with them kind of in our sequencing tools, our notebook, and things like that. As it stands right now, that's not synced to the warehouse, though.
Thank you. You can definitely follow up with Damon after the talks.